Immunofluorescence is the method for spatial tumor profiling, but as with any other scientific or laboratory method, we need to maintain quality to have valuable data. Hi, I'm Alexandra Żuraw, and I'm here to help you do better digital pathology. So if you're up for it, starting right now, be sure to subscribe to the channel and click the bell below to be notified every time I release a new video. In this video brought to you by Ultivue, we're going to be talking about the quality control concept for immunofluorescence and how image analysis and artificial intelligence are crucial nowadays for good QC. So let's welcome Dr. Lakis and dive into the topic of quality control for immunofluorescence. Welcome everyone to the podcast. Today, my guest is Sotirios Lakis, a pathologist from Ultivue. Welcome, Sotirios. How are you today?
I'm doing well, thank you. How are you? Thank you so much for having me.
I am super happy to have you here. The topic of today's video is quality control in the spatial biology environment. Ultivue is a company that focuses very much on spatial biology. Before we dive into our subject, though, let's start with you, Sotirios. Give us a little bit of an introduction to you and to your role at Ultivue.
I'm a pathologist by training, working since 2014, mostly in the biotechnology. And I've been part of cross-disciplinary teams, either doing molecular diagnostics or more recently digital pathology and spatial biology, biomarker development. I started out as a translational pathologist with a little bit of academic experience, but I very quickly switched to the industry and I have stayed there since, although I've also been doing some research. So here I am today, and I joined Ultivue more than a year ago. As a pathologist, I'm involved in many different projects, but my main focus was really the quality control aspects of the product development cycle, because Ultivue is a vendor of conjugated antibodies and kits, and there is a process. This process has multiple QC steps, and of course, the pathologist is very close to quality assurance. I'm really excited to speak about those topics today.
So let's start with the steps. What are the steps? And then let's address quality control for each of the steps.
So even before the steps, maybe we need to kind of distinguish between two types of quality control. If you think about multiplex IF as a workflow, you have different steps. Then you need to speak more specifically about when you're producing something. So when you're manufacturing something, there is also this manufacturing QC. And the interesting part about my work at Ultivue is that I'm really looking into this manufacturing side of things. Mm-hmm. So when you're producing something and you want it to be equivalent and comparable to something you produced before, then you still have QC, and much of this QC is coming from looking at slides, because eventually this is what we call, the functional testing of the markers, where you want to get a certain amount of fluorescence intensity and a certain signal to background ratio. It's more about lot to lot testing than it is about the standard lab-based workflow. However, I believe we can talk about both of these in parallel, because in the end, the QC steps are mostly the same. You start in both of them with the tissue. And I think it's a really important topic, and we can actually spend the whole time discussing about the tissue.
We could have like a conference about this tissue.
Exactly. The tissue as a control, because when we use tissues, we want to control other things. But are we sure that the tissues themselves are suitable for this use? So what we've done at Ultivue is that we have a process for it. And I think most of the vendors actually do that. It's not so unique, but the way that you do it needs to be adjusted to the type of assay that you have and to the type of readouts that you're generating eventually. It's not the same like in DAB, like in Brightfield. So multiplex IF is more complex. So sometimes you have more markers. The interaction between the assay, depending also on the technology, and those things in the tissue that make a difference is slightly different from DAB, because you have different protocols, different ways of visualizing the signal. So you need to take, I think, a very systematic approach in what might go wrong. And then you need to measure this on the tissue. The moment you bring in some tissue, let's say a tonsil that you want to be using as part of your QC, you need to QC it. And I think this is where you need to define what types of parameters are you going to be looking at and what level of confidence you want to have on this tissue. And of course, tonsils are not, we call them normal, but they're not normal, right?
They're never normal.
Exactly.
It's an immune tissue that catches all the threats that come to us from the environment or all the pathogens. So
exactly. So the functional state of this tissue will be very diverse, and you're not 100% certain that a particular marker will return the type of staining you have. Even if you look at the Nordic QC site where they have external quality assurance runs, for some markers, they will tell you that, oh yeah, you will get a staining in the germinal centers, but some tonsils will never give you that staining. These are things that you need to pay attention to, not to mention, of course, pre-analytics. So one of the things we've tried to think is, can we come up with a limited number of markers, like a panel, that captures, for example, these different types of variability that you can get from a control tissue. So the pre-analytical variability, but also the biological variability, so that when you bring in such a tissue, you can have a way of knowing where to assign it. It might be good for your Ki67 or your CD3, but it might not be good for something where you need a certain level of expression to be able to use it as a QC.
That's a really ingenious approach. You basically categorize your tissue and you know which category, or having a different buckets of, okay, this is good for that marker, this is good for another marker, this is good for another marker. And you do this by running the tissue through your process, right?
Yes,
and then
exactly.
The result, okay, so we have the tissue that has to be quality control. Then we have the image. You already mentioned the immunofluorescence image. And then we have the endpoints. You mentioned that already. Endpoints generated by means of artificial intelligence, by means of image analysis. Let's talk about the image now.
You're right that this is a multi-step process. And what one needs to do is take a holistic approach. So depending on the technology and the readout, you will have different types of QC. It really depends on the tissue and on the readout. So there is always an interaction there. For example, with the help of AI, as you said, on one side, you can digest more variability than you could before with the rule-based methods, which could not generalize as well. Even if there is analytical or pre-analytical variability, maybe your system will not be as sensitive. So it will still do a good job. But I think also there is a downside to this. You might be hiding a lot of things inside this deep learning model that works fine, but there might be biases that you don't pay attention to. So I think one needs to really be careful to not introduce biases in this system. Just it happens that one indication is stained with protocol A and another indication is stained with a protocol B, or in site A and site B. And then if you're using deep learning or any sort of machine learning to generate insights from this data, you might find things which are more related to
the batch effect rather than to the biological effect. Yeah, and this is a big issue in the industry, where not always do you have all the information that can cognitively help you identify this bias. Do you have a way to identifying it or to mitigating it?
Yeah, that's a great question. So I think it all starts from data management. People saying that there's nothing in the information that comes with a sample that you should say, oh, that's probably irrelevant. Nothing is irrelevant. So having a way of systematically documenting this information, all the metadata that comes with your sample, at least those that you control internally. We might stain our own tissues sometimes, right? Because we're a company that it has an end-to-end workflow. So we can offer this as well. But sometimes you might not have access to all the steps in the workflow. So you don't really know if there is a bias unless someone tells you. This is where controls are really important, right? Because if you're using the same control tissue across batches, so at least then you have some measure of the variability, because yes, there is inter-slide variability within your control tissue, but it shouldn't be that big. And of course, you can measure inter-slide variability and inter-run variability because they will be interacting in this case. So if you're assuming that you have a tonsil block, which you cap into, 100 sections, and then you're using the 10 first sections for batch 1, and then section 50 to 60 for batch 2, then these sections will be quite far away. I think these are process steps which you need to take into consideration so that your slides are close to each other, or if you have a measure of this variability. So another thing that we're currently doing is, because we're quantitating everything that we're currently staining. So the idea is we have a simple system of quantifying basic statistics, let's say. We're generating basic statistics from the slides. And our idea is that we want to quantify, document, and mine So in this way, you get an understanding that you didn't have before. So a pathologist will tell you, this tonsil suddenly starting becoming dimmer because from section 50 and on, I just see less staining. How much dimmer is that? And how do you know if this is the tonsil or is it probably, the bond? These things become much easier when you use quantitative data coupled with metadata so that you can ask these questions, get the answers, and then modify your workflow accordingly. The steps are important, but the interplay between the steps. So that's what I mean by this holistic approach. And I think sometimes, and it happens in these multi-step processes, that people just give you some data, right? And you have to work with this data. It's really nice if you have the opportunity to see things from the beginning to the end. And I'm glad that I have this opportunity. Let's say in my current role, it's a luxury, I would say sometimes, monitoring everything from beginning to end.
Also, this stepwise approach is something was done classically and still is important. The holistic approach, we didn't really have the tools before. We didn't have the machine learning algorithms, the statistics. Digital pathology was not that widespread. I mean, you are working on digital slides exclusively for quantifying the immunofluorescence. This data was not there anymore. And now this is like an umbrella that can oversee the whole process. That still has to be a sound process addressed in a very systematic way by whoever is doing this. Previously, it was historically, it was just pathologists looking at images saying, oh, this is a little off, but also limited by our visual cognition and visual ability to quantify something, which is not that great. And publications vary on this, but it starts to see the difference when the difference is greater than 25%. That's a lot of difference.
Exactly. If you want more subtle differences, you'll miss them.
So definitely a great QC approach that is holistic and overarching. Okay, so let's talk about the readouts, the quality of basically image analysis that you're doing on the tissue. We already assume everything else is quality controlled. Now we're doing image analysis, AI-based image analysis. How do we QC that?
Every pathologist dreams of a state where spatial biology or spatial phenomics, or people use different terms, does not involve so much of looking at something and having to say, I like it, I don't like it, because it's
Can we reach that ever?
I don't know, but I've also had some experience in the past from the sequencing space for genomics. And what I like there is that everybody says you lose the context. Of course, yes, if you dissolve the tissue into a tube, context is gone. You still have it on the H&E, but it's not the same. So my question is, do we really leverage the context as much as we can for QC, for example? I think there should be a way that you can build synergies between these layers of information that you have on the tissue. So now, for example, that we're able to co-stain on the same slide up to 12 markers and an H&E. This is a lot of information, and H&E is the most stable, let's say, parameter in this equation. So maybe you can use the H&E to do some sort of QC, which is embedded into the system, right? So you don't have to really look at it. The other thing that we will definitely start leveraging in the future is the going from slides to other coordinate systems, because you generate data from the slides, but then you have to somehow visualize them, T-SNE plots or other ways. And this is a completely different coordinate system. So it's not anymore geography, it's something else. So how you treat this multidimensional data, but the future QC should involve a pathologist or a scientist efficiently moving between these two coordinate systems and understanding the data, not always by looking at the slide, but really also looking at the plots and the diagrams.
The plots and diagrams guiding what you're doing and reducing the amount
exactly
of things that you have to look at. I think whoever started early in this space, we were at the beginning of the image analysis algorithms. It was not matching the tissue to the pathologist's liking. So I think many of us developed the necessity, or like, I'm only confident if I have really reviewed everything. But that's not scalable. That's not sustainable with the amount of algorithms that are being run, with the amount of data that we want to generate with them in a short period of time. So definitely moving between, or having a data-driven visual QC process, which also requires training pathologists and tissue experts in interpreting the data, which not always is part of the training. Definitely a good approach as well. Something that has to be future-proof. Looking at everything visually is not scalable, unfortunately.
No, it's not. And if you want to make this a bit more specific, you can say, can the system help me focus on particular areas based on some probability that says, okay, if there is a mistake or if something is off, it's probably here. And yeah, I think pathology is always present, and it's really what is gluing things together, because in the end, the tissue, what we call tissue heterogeneity, which is the reason for success and failure at the same time, is really what makes all these models either very good or not as good. You know, we still sometimes struggle, I mean, as a community, with these high variations in cellularity, for example. You have a packed area with cells, and then you have something where the cells are more scattered. And do we have yet a very good solution to handle these hypercellular areas where the density is like the highest it could be? We don't, I think. However,
No, there is like, even with the basic problem in image analysis, like nuclear segmentation, there are better and better models, but visually you cannot distinguish it. You can maybe project, you can maybe derive information from the sizes the cells should have, but I have not seen 100% reliable solution so far.
No, and maybe we don't need 100%. The reason I brought this up is because you need to check these areas. So if something is going to fail, it will not fail
That's
equally.
where it's going to fail.
Exactly. That's how pathology can be introduced into this QC process, but it doesn't have to be necessarily a pathologist looking at it and saying, yeah, I like it or I don't like it. You have to take what can be extracted from a pathologist and turn it into something smart that is automated and helps the user navigate on the slide efficiently and really spot the areas where something might have failed. I think it sounds good, I would say, but in the end, sometimes you just have to look at the slide, right? So you need to be
there. You need to be able to troubleshoot. As much as we're involved in digital pathology and we want to think this is mainstream, it's not mainstream yet.
No.
It's still cutting edge, and it still has certain challenges, and quality control is one of them. Thank you so much for walking us through Ultivue's process, through the stepwise and the holistic approach. I hope it's going to be useful for everyone who's watching it. And thank you so much. Have a great day.
You too. Thanks a lot.
There is no reliable data generation without a good quality control process, and Ultivue is very much prioritizing QC. If you want to learn more about Ultivue and their approach to tumor spatial profiling, be sure to visit their website at ultivue.com, or you can click on the link that I will include in the description below. And I talk to you in the next episode.